embryonic development in Drosophila. And what I showed you was that the segmentation genes control the homeotic genes. They do that by combinations and concentrations of gene regulators, transcription factors. So that's the specific system for insects, but not even all insects. So many different species, there are variations on theme. But you have a similar but not identical pathway working, and your homeotic genes are called HOS. And they essentially do the same thing. They give identity. You don't have segments in your body plan, but as I pointed out on Monday, you have segments. If you run your finger up and down your spinal column is segmented. And so that original function that used the Drosophila homeotic genes to segment the body plan and the nervous system is used for you to segment your nervous system. You use a different set of genes to segment and identify anterior versus posterior, dorsal versus ventral. And so we're going to make the switch now to mammals, and specifically humans. The basics are still the same. That's why I want to emphasize that what we're going to pick up now is merely a continuation of what we did on Monday. Even though what we did on Monday was in an insect, and some of the genes are not going to be present in your developmental system, the basics are still the same. We have sets of genes turning on and turning off other genes. And what's really important about the homeotic genes, whether it's the Drosophila homeotic or human homeotic, what's important about them is that they become epigenetic. We've talked about that before. We've talked about it in terms of in females, when the Barr body chromosome is identified in each and every cell, the memory of that, whether you're making a Barr body out of your mother's X or your father's X, that memory that's incorporated into the chromatin protein structures of the chromosome. So the exact same thing is happening here in flies and in you. When segments are identified early in development, that identity is enforced in the chromatin epigenetically. We talked about that earlier. The definition hasn't changed. It's a modification to the chromosome, basically the histone proteins, and every replication cycle, that modification is reproduced. And so, for example, your nerve genes, the genes that make nerve cell proteins, are in open chromatin in your nervous system. They're in closed chromatin in your blood-forming cells. And so that's what's important coming out of here. Because we're now going to switch to your cells and look in bone marrow. And the system that we're going to look at, the two systems that we're going to look at, is first of all, your red blood cells and the protein globin. They form in your bone marrow, and white blood cells, which make antibodies. And so the system which identifies bone precursor cells is just a continuation from the homeotic genes that identify what different tissues will become. So even though we're skipping ahead in time and we're skipping species, the process is really comparable. And so what we're going to be looking at first is red blood cells and how globin genes are regulated. And then we'll switch to white blood cells and how antibody genes are regulated. Just again, to reinforce this concept, globin genes are open in red blood cells. Antibodies are open in white blood cells, but antibodies are closed in red blood cells. Globin genes are closed in white blood cells. And that open and closure is enforced by epigenetic modification of chromatin. That's what this is all about. That's what we're going to be looking at. Yes?
Does closed mean that basically red blood cells, the antibodies are always the same, as opposed to variation in white blood cells?
So what I mean by open and close, remember we had the different forms of chromatin. So beads on a string, that's open chromatin, as opposed to the 100 angstrom and 300 angstrom fibers, the folded up beads on a string. That's a different form of chromatin, a different structure. That's what we are talking about. That's what open and closed chromatin is. So genes that are going to be used by a cell are typically in open chromatin, and the memory of that is fixed in the histone proteins. Anybody else before we get into this? All right. So we're going to look at red blood cells. Red blood cells are the cells in your body that pick up oxygen in the lungs, circulate throughout the body. The oxygen gradient drops outside the lungs, and so the red blood cells deliver the oxygen to all the cells throughout your body. It does that because the globin proteins are in a structure called hemoglobin. Hemoglobin contains iron, it contains the steroid heme, and it contains globin proteins, 2 alpha type and 2 beta type proteins. And that's what is going to be our focus. Our focus is actually going to be on one part of this system, and that is the beta type globin protein genes. So hemoglobin is a tetramer, 2 alphas, 2 betas, so that's 4 globin proteins. It is a double dimer. So 2 of each, a dimer of alpha type and a dimer of beta type. So that's very simple, very straightforward. That's what's in the red blood cells. So there are alpha type genes and beta type genes. They're very similarly regulated. We're just going to look at the beta type. But that's only part of the problem with mammalian cells. If you think about it, what distinguishes mammals from other vertebrae are the fact that mammals have a placenta. And so as the embryo is developing, the embryo can't get oxygen from its lungs. Very young embryos isn't even going to have lungs yet. So the embryo has to be able to grab oxygen from the mother. And that's a biochemistry problem because the mother, her red blood cells are designed to deliver oxygen to tissues. The embryo is trying to grab oxygen and deliver it to its tissues. So the way that evolution has solved this problem in mammals is that when you were an embryo and when you were a fetus, you used a different set of globin proteins. So the alpha and beta designation, we'll elaborate that in a few minutes, that's the adult set that you all are using right now. But when you were an embryo, when you were a fetus, you had to have a set of globin proteins that was strong enough to grab oxygen from the adult hemoglobin and then deliver that oxygen to the embryonic or fetal tissue. And so that required a very, very different and more elaborate set of globin protein genes. So what we are going to look at is a group of genes grouped together where we have first, we have an embryonic gene, then we have a fetal gene set, and then we have an adult set. And so the development and the regulation of this is that the embryonic gene turns on first, and then it turns off, and then the fetal set of genes turns on, and then they turn off at about the time of birth. All the hormonal changes occurring when women start to have labor will turn off the fetal genes. All those hormones will then activate the adult genes, and there's a handover. So that's the big picture where we're going to go with this. Any questions on the background for what globin is all about?
So, the fetal globin, how long does that stay in effect after birth? Because
So
those red
there's,
blood cells are going to be around after birth.
no, and that's absolutely true, but it doesn't matter. So you're right. It's very individual. So it can be days to a few weeks. But then there's a mutation called persistence of fetal hemoglobin, where it never turns off. And those people are perfectly normal. You never find out that an individual has that until they have blood work done. So it's not the end of the world to have the fetal hemoglobins left on accidentally or on purpose.
So they'll transfer O2 just fine?
Yep.
Throughout? Okay.
Yep, and they'll pick it up better and transfer it just fine. Anybody else in the background? Yeah.
So is there an evolutionary advantage to switching to adult hemoglobin after birth?
That's an interesting question. I have no idea. I don't know. I've never thought about it before. Because the fetal persistence syndrome is absolutely without phenotype. Anybody else? All right. So the alpha type genes and beta type genes each are located in one cluster each on different chromosomes. And so we're going to focus on the beta type, where we have beta and delta genes. So you actually have two different adult type globin genes. They are virtually identical. There's only one or two amino acid difference. They were formed by gene duplication and then one mutated slightly. They are almost identical. So each one of these boxes is an individual transcription unit. with a promoter and a terminator. I'll give you the gene structure, the intron-exon structure, in a few minutes. The major regulator is at the 3' end, and so this is an enhancer. So the regulator for the globin genes is not located at the 5' end of the gene. The regulator is located at the 3' end. That really doesn't matter because most of your regulators are located a long distance away, and they all act by the DNA looping over, almost like a pretzel, to contact the promoter. So enhancers literally can be anywhere, even inside a gene in an intron. It doesn't matter. In these genes, they just happen to be in the 3' end. So this is your adult set. Here we have gamma 1 and gamma 2. Again, independent transcription units, enhancer at the 3' end of the gene. And then out here, we have epsilon. I think I told you the first day of class, you become fairly well versed in Greek. And so these are all Greek letters. This is fetal and embryonic, promoter, terminator, and enhancer. And so those are your active genes all in one cluster. There's another gene in there, which is a pseudogene. And that Greek letter is psi for pseudogene. It's located there. So you really have hundreds to thousands of dead genes littering your chromosomes all over. A pseudogene, classically, is a gene whose gene structure suggests that it cannot function. That's not entirely true anymore, since we now know that almost everything is transcribed, and lots of pseudogenes have functions. They just don't have the same original function. And so there's a pseudogene there. It may not be doing anything in this cluster, but that's not true for pseudogenes in general. So here we have the time schedule from early embryonic, we turn on epsilon, then we turn it off and turn on the gamma genes, they turn them off and turn on delta beta. Beta is your major, delta is your minor. If you have blood work done, you will have alpha beta as your major hemoglobin and alpha delta as your minor hemoglobin. Excuse me, any questions on that so far? And then out here, we have the globin locus control region. So the globin locus control region is a ginormous, huge, multiple enhancer. It's very, very large, and it regulates this whole set of genes. So we have two different levels of control for your globin genes. One level of control is at the individual gene. So each individual gene has its own individual regulator. The globin locus control is like the master regulator for the whole region. So again, the big picture, the way this works, is that what we will do, what I will show you, is that very early in embryonic development, you're going to want to turn on the embryonic genes. But the way we do that is to open the entire region, and the globin locus control will pick out the embryonic gene to turn on. And so that's the big picture for how we're going to start. Now, this is the time sequence, and so we're going to turn on embryonic first, then fetal, and now you are running your adult genes. These are all turned off. There's one more wrinkle to this, and that is not only is there time course regulation, but there's tissue regulation. So this is done in bone marrow. This is done in the fetal liver. This is done in the embryonic fetal. This is done in the embryonic yolk sac. And so not only do we have regulation over time course, but every time we change the use of the genes, we also do it in a different tissue. Any questions on this so far? All right. So again, I want to emphasize one thing on the top board for 1 second. That's the paradigm. That's what's all important, is that development is constantly changing. As we go from early embryos to late embryos, as we form anterior, posterior, dorsal, ventral, tissues come on, homeotic genes say that this is going to be a thorax, this is going to be a head, and then within the head, other genes turn on and say, well, this part will be an eye, this part will be a nose. So that's a constantly changing set of regulatory events. And so what that means is it's all run. Change means that the concentration of individual regulators and the combination, so some are going to turn off with time, and as time marches on, just like we saw in the early embryos, we went from the maternal genes, the bicoid, the nanos. We went from the maternal genes to the segmentation genes. I didn't give you their names. They're in your textbook. Those got turned off, and we went to the homeotic genes. And so that progression is just going to continue right through adulthood. And so as we start switching over from homeotic genes, in the yolk sac, we turn on for the concentration and combination of gene regulators is perfect in the yolk sac to open this up. And what happens is that the globin locus control concentration and combination of transcription factors becomes perfect in the yolk sac to activate the globin locus control region. And so in the yolk sac, this whole region opens up. The entire region. But the combination and concentration is perfect for the GLC and for that enhancer, which then turns on that gene. And so in the embryo, in the yolk sac, we begin making the epsilon gene And the same thing is happening on a different chromosome. We're making one of the alpha type genes. And so we're making the first of the hemoglobin proteins. So now the embryo is competent to grab oxygen at that blood barrier in the placenta to begin feeding oxygen on its own to tissues. Everybody see how this works? The whole region is going to open up. Any questions on that? Yeah.
Just to clarify, the GCL is what starts each enhancer at a particular time.
Right, so that's the master regulator for the entire region, but it knows which gene to hit.
So at each time.
At each time point, in each tissue, it knows in the yolk sac to turn on the epsilon gene. You got a question?
Yeah, does that say open, does that word, what else the word say about
Concentration and combination, over and over and over again. Concentration and combinations of transcription factors that are always changing as development progresses. Individual regulatory genes will turn off, other regulatory genes will turn on as time progresses. Any questions on that?
The DLC turns on the yolk and the oxygen?
Anybody else? So, as time marches on, the concentration and combination of transcription factors will change until in the yolk sac, it's no longer the proper combination and concentration to run this system, and this will close, and instead, with time, the combination of concentration in the yolk sac changes, it turns off there, and it turns on in the, we now are further developed, we have a fetal liver. In the fetal liver, the combination and concentration becomes correct. And now we open up the whole region again. The whole region opens up, but the GLC is now recognizing these two enhancers because the combination and concentration of transcription factors that bind to these are the correct ones. It doesn't recognize those enhancers. It doesn't recognize that enhancer. And so now the entire region is open, but the gamma genes are specifically turned on. Time marches on. As time marches on, we are going to close down the system in the liver, and then shortly before birth, the combination and concentration of gene regulators in the bone marrow cells becomes appropriate. The GLC shuts down in the liver and opens up in the bone marrow, and now that is what you are running on now. And so you will use this system how this works. So each one of these genes genes are identical in structure. All the genes, the alpha and the beta genes, came out of a single gene that duplicated and diverged. And so that's going to be a common theme towards the end of the course when we hit the chapter on evolution. Gene duplication and then random mutation, selection for good function, is what allowed the development of placental development. There could not be placental development unless there was this multiplicity of globins with different oxygen binding abilities. Any questions on how this system works? So now, we are going to focus on the second part that's coming out of the bone marrow. So there are dozens, perhaps hundreds of different white blood cells, all having a common origin. We are going to focus on one specific type called B cells that make antibodies. So white blood cells, the red blood cells have a common origin in terms of the tissue that gave rise to them. Antibodies are the proteins that you make. They have specificity for antigens. That's the other part. So antibodies' function is to recognize a structure, sort of like a hormone receptor. Now when antibodies recognize a structure, they bind to it, and if that structure is a bacteria or a virus, it leads to its destruction. If it's a pollen grain, it leads to your allergies. So it's the same system working the same way. In a sense, antibodies are kind of similar to the globin in that we're talking about a tetramer of 4 proteins, a double dimer, 2 of this and 2 of that. So the general antibody structure looks like this. We have 2 light chains designated L's. Those are the small proteins. 2 heavy chains designated H. Those are the large proteins. My proportions aren't exactly correct. The heavy chain is 4 times the size of the light chain. So we have a double dimer, 2 L's and 2 H's. Unlike the globin, where there is no covalent attachment, these are all covalently joined by disulfide bonds. And so that's what the SS is. Those are cysteine amino acid residues. They're 2 sulfur-containing amino acids. Cysteines form covalent bonds fairly readily. And so the antibodies are a tetramer, a double dimer, held together by disulfide covalent bonding. Not true for the globin, they're held by hydrogen bonds. Now, what's interesting about antibodies is the fact that you have about 20,000 plus genes. Yet you can make tens of millions of different antibody proteins. And so going back to the 1950s, 1960s, that was one of the great mysteries, is how an organism with a known limited number of genes can make a virtually unlimited repertoire of antibodies. Someone could sit down in the laboratory and design an organic structure, inject it into you, or more commonly a goat or a rabbit, and within a week to 10 days, that structure will cause the B cells to start producing an antibody which will lock a key, lock onto that structure for its ultimate destruction. So again, it was known way back in the 1950s that you can make an almost unlimited number, millions and millions of antibody proteins from a limited number of genes. And so that was very mysterious until people finally entered the cloning era and began to see the structure of the antibody genes. The mystery was solved. And so part of that mystery has to do with the structure of the protein itself. We have amino and carboxy end of the protein. So we know this terminology, we've had it before. The protein has an amino end and a carboxy end. The red line through there is a line that people identified, I guess, late 60s, as they began to sequence the proteins and were able to get clonally pure antibodies. If you go into your body, you can't get clonally pure antibodies unless you have leukemia. And so leukemia is a white blood cell cancer, soft tissue cancer, where one antibody-producing cell begins to expand uncontrollably. And so, because one cell is clonally expanded, people were able to get from leukemia patients, each leukemia patient made a completely different antibody. And so as people began to sequence the antibodies from leukemia patients, they discovered something very interesting, that they could literally draw a line through the antibody protein, the tetramer, which cuts the light chain about in half and the heavy chain about in quarters, and identify a variable portion and a constant portion of the protein. So that was the first, something unusual. As you look at hundreds of different antibodies from different leukemia patients, what you saw was they all had, almost all had, the same or very, very similar constant part of the gene. The back half of the light chain, the back 3/4 of the heavy chain. But every single antibody had a different amino half, again, on the light chain. And so that was the first clue as to how you can make millions and millions, unlimited number of different antibody proteins. Any questions so far? I've been going on and on. Okay, so that was in, I guess the late 1960s. Then the next big surprise, as people began to try to map where the antibody proteins originated from, to map the genes, they discovered something variable portion of the gene was coded for separately, genetically unlinked, separately from the constant portion. And for a while, it looked like two genes coded for one protein, which was just heresy at the time. So we have a separate coding part of the chromosome for the variable part of the protein, separate from the constant part of the protein. And so that's where we're going to pick up the story. As people began to clone and sequence the antibody genes, the immunoglobulin genes. Let me put that word up on the board. So this is immunoglobulin as opposed to globin. Please do not mix up those two words. If I ask for one and you give me the other, I won't give you any points. Make sure you get that terminology correct.
When you're saying that you said that they code for the same thing, are you saying they code for the exact same thing or different parts type of thing?
So the constant portion, what we see is the exact same constant portion connected to hundreds, thousands, millions of different variable portions in the protein.
But no, yeah, when you said the thing, it was heresy that two things code for the same.
So the heresy is two genes coding for one protein.
Okay.
So the dogma back then was one gene codes for one protein. But now we seemingly have a situation because the variable coding part was genetically unlinked from the constant coding part. It looked for a while like two genes coded for one protein. Anybody else? Okay. So the answer came in looking at the DNA sequence of the antibody region. And so we are going to look, let me label some of these. So we have kappa or lambda. There are only two different light chains. We are going to look at kappa. For the heavy chains, there's alpha, gamma, M, let's see, and a couple of others. The heavy chain gives its name as IgA, IgM, I think it's IgG that people with asthma have a problem with. So the name of the antibody comes from different heavy chains, an A chain, an M chain, a G chain. Connecting to either kappa or lambda. We'll go through the DNA first, and then we'll review the biology of how this system makes antibodies. So again, any general questions on the terminology before I show you how two different regions, genetically unlinked, can make a single protein? So two different regions, genetically unlinked, but on the same chromosome. So the kappa genes are all located on one chromosome. The lambda genes are all located on a different chromosome. And the heavy chain genes are all located on a third chromosome. Each one is done independently. We look at the biology at the end. So what we have is a set of variable region I like to call them cassettes. They're really not genes. They are part genes. I'll explain that as we finish up the drawing. Cassette is really a good word to describe what's going on. So we have the body of the variable portion, the front portion. Remember now, these are the kappa genes. The variable portion comes in two exons, L1, V1. The space in between is an intron. And we have L2, V2, L3, V3, L200, V200. Each one of those V regions, and so the whole thing is called the V region, but the V region is in two exons with one small intron. Each one has its own separate promoter. No regulator, that's really important, no terminator. So it's not a transcription unit. The transcription unit has a promoter, regulator, and terminator. All we have here is a promoter. And then out here, we have the kappa constant region. It's got a transcription terminator. And here, takes me a second to draw all these. We have joining regions, J1, J2, J3, J4, J5, and J6. The joining regions are little mini exons. And then, in the intron in between J6 and kappa constant, we have the regulator, the gene regulator, genetically unlinked to the promoter. So the way this stands, you have two separate gene regions on the same chromosome. We have a set of cassettes with promoters that can't be used because there's no regulator to turn them on. And we have the regulator and the other half of the gene and the transcription terminator half a chromosome away. Not exactly half, but long ways away. Everybody see? This is the paradigm for all three genes. Remember, there are three different ones. There's lambda and kappa light chain, and then we have all the heavy chain. They all work about the same way. Not exactly, but similarly. So we have a problem. The problem is, to make an antibody protein, you have to take a variable portion and connect it to the constant portion. The variable portion will bring its promoter with it. The constant portion has the enhancer and the terminator. Any questions on the structure?
So the B's don't have promoters or terminators. The constant region only has a terminator and a promoter, just an enhancer.
Right. This is called the germline gene sequence. So this is the sequence, this is human. This is the sequence of your kappa light chain antibody genes in every cell in your body except your B cells. So there are hundreds of different white blood cells. The B cells specifically is going to undergo, let's use we're gonna close our eyes and select the V. We close our eyes and select a J. And do site-specific recombination. So we're going to take randomly the end of any one of the V's and the beginning of any one of the J's and literally do site-specific recombination. Remember, Lambda used that term for integration into E. coli, site-specific recombination. It's not using the general recombination system. It's using a very special recombination system, special for this system and one other one. And so we're going to go from the germline sequence, and so everything in between, all that DNA is going to be thrown away. Big chunk of DNA. But it's only in this cell and all of its daughters. And so this is going to give us L2, B2, promoter, J4, J5, J6, and so now we have antibody gene, a light chain antibody gene. Notice there's a gene that actually looks kind of like globin. So it's a gene with 3 exons. The first exon is the leader. The second exon is the VJ exon. The third exon is kappa constant region. We now have a real, complete gene made from 2 distal segments, 2 cassettes that we put together by recombination. We have a promoter, a gene regulator. It's in the intron, but it doesn't matter. And a transcription terminator. And so now this gene can be activated and fired up. And we can start transcribing it and make an antibody. And the same thing is going to happen, but in a more complex way. Look in your book to find the details for the heavy chain. Heavy chain is probably 3 or 4 times more complex. But the basic mechanisms are exactly the same. Yes? Oh, J stands for joining. Thank you. I should have mentioned that. So J stands, those are the joining segments. That's what the V joins to the C, but it doesn't really join the C. It joins to a J region. And so this is the gene structure that you find in your circulating B cells, that population, that small proportion of white blood cells circulating along with the red blood cells.
How come like J5 and 6 got to stay along, but everything from B2 to 200 didn't stay?
The thing was that everything from there to there eliminated. Now, J5 and J6, you see, are in what we now call an intron between the VJ exon and the C exon, but they're just sequences that didn't get used. They're in the intron, and when this message gets transcribed and spliced, it just gets thrown away. There's nothing else to do with them. Whatever is in between that's eliminated, if we had used J1 and we have a larger intron with 2, 3, 4, 5, and 6 J's in the bigger intron, but it would still get spliced out so it doesn't affect the final product.
What is the gene labeled between the promoter and V2?
I didn't get that.
The gene, like what is it labeled between the promoter and the alpha?
L2.
So we have L2, V2, J4, and then C kappa. Is that what you're asking?
Yeah, I was asking for the alpha.
Okay.
Yeah, thanks.
Why is that one B one not on there? Oh,
I just elected not to.
but it doesn't use it?
No, that's right, it doesn't use it, because this enhancer is going to control the nearest promoter. And so you're right, I mean, it is definitely out there, but it's non-functional. Anybody else? So we are getting close to the answer to the big question, how do you make tens of millions of different antibody proteins from a few hundred? You've got about 100 or so here, you've got another 100 or so in the lambda, and you've got 100 to 150 in the heavy chain. And from those few hundred, you make millions, tens of millions, uncountable numbers of antibodies.
What is the
Leader. L stands for leader, V stands for variable, J stands for joining, C for constant. Those are the major those are the parts of the kappa light chain gene. Lambda is very, very similar, a little bit different organization. Anybody else? So, immunoglobulin diversity. How do we get tens of millions, even hundreds of millions of proteins from this and lambda and heavy chain? First of all, we have multiple variable regions. Secondly, multiple J's. So those will multiply together. If we have 106, we have 600 different possible combinations.
So the J's like J exon, yes?
Well, from the no, not really, because when the recombination happens, it gets tacked onto the V, and so it's called the VJ exon. So a J is never a standalone exon. Anybody else? So one way in which we make millions is by multiple V's and multiple J's. Now the second way is if we look closely here, let me bring that down. If we look closely at that left joint of what will become the VJ exon, the reality is we don't just cut it at the end of the box, but the way the system cuts is It can cut there, or there, or there, or there, or there. And so what we have is called imprecise excision. And that happens on both ends. So imprecise excision on both ends means that if you cut, let's use that as a starting point, if you cut there, you have one sequence, that's a sequence in the open reading frame. But if you cut one base before or one base after, that's a real frame shift potential. You're changing the reading frame. That's in essence an insertion or deletion of one base. And so because of that, we have this imprecise excision there, and I'm not going to draw it in, imprecise excision on J4. And so that's going to give us a variable readout of amino acids. The reality is that the blue box is where antigen binds. So the binding is going to be right at that end of V, into all the J, and into the beginning of C. And so, if you can visualize now, we're going to have this tremendous diversity because of imprecise excision. But that's not all. Because the next thing that happens is called nibble add. So I already described the nibble part. Add means that we're going to randomly add nucleotides. So an enzyme is going to come in once the cut is made and the end of this segment of DNA is now open. Let's say we open it there. An enzyme is going to come in and just randomly add a few nucleotides. To both ends. Both ends are being treated exactly the same way. And so again, it's this region that encompasses that red line. The red line is exactly where we made that site-specific recombination. We nibbled out a few bases randomly. Could be one, could be two, could be three. And then we added a few random nucleotides. 1, 2, 3, 4. With every one, you're going to get a different protein, a slightly different protein. It's uncountable, the number of proteins that you can make. Because we're not done with why is there diversity yet. So this is just the mechanism of what's going on to make a right chain. Any questions on that? Next we have multiple combinations of light chains and heavy chains. And so each cell will only make one light chain. do you see the problem in this system if you're randomly adding and subtracting nucleotides in an open reading frame? Yeah, it might make a stop codon, that's exactly right. And it happens. And if that happens, this is the first thing that goes. If you make a stop codon and make no protein, the cell will automatically switch and do the same thing to lambda. So kappa is the first choice, lambda is the second. If the same thing happens to lambda, the cell will undergo programmed cell death. It's no use. You can't make an antibody, it's all it's good for. The cell will die. If you make either lambda or kappa, now you have to have a good heavy chain. Same thing happens at the heavy chain.
Is producing a stop codon very rare?
I think it's probably pretty common. I think it's probably pretty common. But there's more of a problem in that not only can you make stop codons, you can also make self-antibodies, antibodies against self. And so this whole thing is set up so that you have to make a good antibody. I don't know that we're going to have time to go through the biology. We picked that up on Monday. But this whole thing is designed to make an antibody, which then gets filtered out. Because the cell can't afford to make a B cell and throw it out into the circulatory system in case it's made an antibody against you. But this is the way the system works. Now, the heavy chain, you look in your book, we're not going to do it. If you look in your book, the heavy chain is twice as complex. So the light chain only has one site-specific recombination. The heavy chain has two different site-specific recombinations. Either one can lead to no reaction, a stop codon or self, and get eliminated. This is all happening randomly. Now, the big mutation in this system is the recombination system. It's called RAG. The RAG system. People who have mutations, homozygous mutations, these are the children in the bubble. These are children that can make zero antibodies and have no means of fighting off infections, and they have to be extraordinarily careful. Most die of infections at a fairly young age, just because they can't make antibodies. But this system is designed to give maximum variability. And the variability is right in here due to all of those things. The exact same thing happens in heavy chain, but it just happens twice. Any questions on antibodies?
What's a drag cell?
I was hoping nobody would ask that. I don't know. You guys know what RAG stands for? I don't know. Okay. Someone will Google it. So this is the mechanism. Now, the biology of this system is such that this happens every day, every minute of your life. Your red blood cells, the hemoglobin, yeah. Recombination activation gene. Thank you. I knew somebody would do it. Okay? Those recombination. This happens every day. And the cells have about a month lifetime. And so every day you are randomly making millions of different antibodies. And every day millions of different B cells are dying. You cannot sit down and think, I'm going to make an influenza antibody tonight. That's not the way the system works. The system makes antibodies randomly, and if one happens to hit an antigen, that's going to be clonally expanded. And so what that looks like all of the antibodies are sitting out on the cell surface. Remember, each cell is only allowed to make one antibody. It tries kappa first. If it makes a good kappa protein, fine. If not, it goes to lambda. And it only gets one shot at H. There's only one H locus. And so if we have good antibody, it now decorates the surface. This cell has to go through a self-filter. If this cell reacts in the filter with your antigens, with you, that will trigger programmed cell death. And so I think the estimate is somewhere around 50% of all the random cells, antibody B cells, are going to die because they react to cell. Only those that don't get out into circulation. And now this cell is going to circulate for about a month. If it meets an antigen, and we bind an antigen there, that's going to trigger signal transduction. Very similar, but not identical to the growth factor signal transduction pathway. The signal transduction pathway, remember, is the key in going from actually G0 to S. Not G1. The cell is not in G1. It's in G0 in a resting condition. And now, it'll take a few days. Hopefully that's enough time to bind out all the antigens. Viruses multiply very fast, so do bacteria. And so there's a race. After the cell has cleaned up the antigen, all the antibody cells that were amplified, a small group of them are going to be selected as memory cells. That's what immunization is all about. So if you were exposed to anything, you have an immunological memory of that because some of those antibody cells that reacted are going to be held out special and put in reserve. For reasons that aren't clear, some antibody memory lasts a lifetime. Others you need boosters before it decays. That part's not clear. Any last questions on this? Yes?
Anderson?